Can you hear me? Good. Good. So sadly, this is the last lecture, and I realise I won't be able to cover all the things I promised, but we'll see. And I realise I have to finish by 11 because there's a group photo. So that gives you all hope, I think. So I'm going to talk about stabilizing selection, but really about, I think, one of the big questions in biology, which has had a rather strange history. Why is there such high heritable variance in all kinds of traits, including traits related to fitness? And of course, this was a dominant theme, as we heard, in Darwin's theory, in The Origin of Species. He spends a lot of time documenting the existence of high heritable variation. And strangely, even though through to the sort of mid-60s, 70s, there was a huge debate over the causes of molecular variation, which had been discovered in the mid-60s. And maybe that just sucked away all the energy from people to think about things that actually matter, the traits that we really care about. And people didn't really start to debate why there was such extensive heritability until really the 1980s, when there was a kind of big argument, particularly between Russ Landy, Michael Torelli, and others, trying to understand how we could explain heritability. And then that kind of faded away, I think got a bit diverted again by the rush of more data, QTL mapping, now GWAS. And coming back in the human genetics literature in trying to understand GWAS, trying to understand the distribution of effects on traits versus allele frequencies, which you've heard about in several talks, all of that. And so people are now focusing again on maintenance of variation, often without very much literature, very much connection with the earlier literature, which I think Bruce mentioned. And also very much focusing in the classical sort of framework on the explanation which has received the most attention, which is that heritable variation is a balance between mutation introducing variation, and we've heard it's introduced at a certain measurable rate, quite a high rate, versus stabilizing selection, favoring an optimum and disfavoring individuals that deviate from that optimum. And so it is, in the sort of classical tradition, a mutation selection balance, with variation being essentially deleterious due to deviation from some optimal combination of traits. So what I'll do is start by sort of summarizing things that we know, and important things, some things we know pretty well, some things we have a very vague idea about, and some things I think we can't even define very clearly. So I'll go through what I see as the facts, and no doubt there can be a lot of argument about my attempt to summarize this. And then I'll talk about the sort of theoretical framework, contrasting different models. And the main point I want to make, which I think I will succeed in is that it's all very complicated. That although mutation selection balance seems very clear cut, there are actually a variety of models, and even though the phenotypic quantitative genetics is very straightforward and simple in some ways, details about the underlying genetic basis make a big difference to how much variation we see. And it's really quite difficult to get at those details. So it's kind of frustrating that we have this very successful quantitative genetic framework described really by the infinitesimal model, very successful in practice. And we know a huge amount about molecular variability, but connecting the two is really hard, okay? So, observations. Well, as we've heard, there's high heritability for most traits in most sexual outcrossing organisms, and this variance is largely additive. What effect does it have on fitness? Well, one can, in principle, very straightforwardly measure the selection on traits by measuring fitness or some proxy for fitness, survival, mating success, or whatever, as a function of the trait value. And very noisy, very difficult to do, but if you do a reasonably big study, you can then do some kind of quadratic regression of fitness against, or fitness proxy against trait, and you may find a sort of negative quadratic relationship with a certain curvature, which we approximate typically by some sort of model of stabilizing selection, which is that fitness simply decays in a sort of Gaussian model around some peak, okay? And so we have an optimal value, we have selection pushing populations towards that optimum, and the crucial parameter is Vs. And the square root of Vs is, if you like, the standard deviation of this curve. It's a measure of the range of trait values which are reasonably fit. And another way to think about this is to see that the mean fitness is reduced by an amount which is basically the mean squared deviation from the optimum. And if the mean is at the optimum, then that's going to be reduced by genetic variation around the optimum, Vg, divided by 2 Vs. So one way to think about this Vs is that the larger the value, the weaker the stabilizing selection, and the loss of fitness due to genetic variation around the optimum is just Vg over 2 Vs. Okay? That gives you a sort of measure. So that's all straightforward, and there were a huge number of studies really triggered by a paper by Landy and Arnold showing how one could measure multiple traits and actually estimate not just the relation of fitness to this set of traits, but actually estimate the effects of particular traits on fitness, accounting for covariance. Okay, so accounting for correlations amongst traits. And this was actually an extension, really, of a practical application of a framework developed by Carl Pearson, really in around 1900. So it's quite an old method. A little bit misleading in a sense, in that although one can account for correlations between the traits one measures, you can't measure everything. So that is a point I'll come back to several times. At any rate, there were a huge number of studies of strength of stabilizing selection on traits. And these, I mean, it's maybe, the Landy and Arnold paper provided a research program, a straightforward research program. You go out and you measure a lot of organisms. You measure survival, let's say, you compare age classes, or you compare mating pairs with non-mated, etc. Measure traits. You get a PhD thesis, right? So there were thousands of studies. There was then a rather sobering meta-analysis by Kingsolver et al. 2001, which showed that yes, you do typically get strong selection estimated this way. You find strong, significant relationships between traits and fitness. And these often gave a significant quadratic term, a curvature, but disturbingly often the curvature was sort of positive instead of negative. So we had that kind of relationship, which would imply that the traits would go off to infinity in one direction or the other direction. Another somewhat disturbing thing that came out of this meta-analysis is, it's called the funnel plot. If you plot the sample size against the value, and gamma is this curvature value here, let's say, I can't remember which way, is positive stabilizing selection, on which convention shall we use? Anyway, let's say this is stabilizing selection, and this is disruptive selection. So you get a whole lot of values, as I say, some indicating disruptive selection rather than stabilizing selection. That's a worry. You get these rather strong values here. But the disturbing thing is that the larger the sample size, the weaker the effect. And that really suggests a pervasive publication bias, that people do lots of studies and they can publish the big significant effects, or they do multiple studies and they stop when they get something significant or whatever. Various kinds of unconscious bias or even conscious bias. And this really means that I think we can say that many traits just naively have some strong relation to fitness. People are often measuring necessarily interesting traits, but it's very hard to say anything quantitative. Okay. So I dare say the last word is in the 2018 book. But I think it's still true that this is a very murky area and very frustrating to put so much effort into measuring something and not quite knowing what's coming out of it. Anyway. Yeah. Which, the picture? This plot? Okay, so this plot is just my attempt to say, each dot here would be a study, someone's PhD thesis, right? And it's an estimate of the as I plotted it, of the curvature of the relationship between trait and fitness. So if you see, you know, that what one would expect to see is some nice curvature like that, indicating selection towards some optimal value, and that would be a point up here. That's great. But if you plot all of these, you find there are quite a lot of studies which point to the opposite pattern, which is kind of impossible in the long run, because this would not be an equilibrium. And you find that studies which have a larger sample size give you smaller values, right? Presumably because, you know, if you say, well, I'm going to publish when it's significant, you can get a smaller effect will be significant if you have a larger sample size. That's all. Yeah. Yeah, but has there been a meta-analysis more recently comparing the new methods with the old methods?
Not really.
Okay, yeah. I don't really want to dwell on this. In fact, what I'll say in the end is that regardless of all the experimental evidence, I think it just has to be the case that in the long term, most traits or many traits, functional traits that affect fitness have to be under stabilizing selection, because we observe from the fossil record that things morphologically, for example, stay very, very similar over very long periods of time, and traits which affect fitness stay very constant.
So it presents 6 lines of evidence.
Okay, I won't go on anymore. Yeah, okay, so we can have more argument later. Yeah. So I'm just going to assume stabilizing selection because of a lack of imagination of how else could it be. And just to mention that, you know, we'd like to have some, you know, decent estimate of the strength of stabilizing selection, this VS term. And traditionally, this goes back to Russ Landay, I think, in his early papers, it's taken as 20 times VE, meaning that the sort of standard deviation is, you know, 4 or 5, 4 point square root 20, as big as the non-genetic variance. You know, so that corresponds, if we look at this VG over 2VS, to a loss of fitness due to genetic variation of about, what would it be, 25, no, what's it, 20, 40, 2.5%, if we get that right, 2.5%, 1 over 40, is a sort of loss. So, you know, that's a sort of modest selection, which is believable anyway for some trait. And this is just taken as some number to put into the theory. So I'll use that, but treating it really as a conventional value people use. Well, I mean, stabilizing selection, of course, implies epistasis at the underlying gene level. If we think of an additive trait, I haven't actually assumed that, but if the trait's additive, then, you know, a plus allele would be good if you're below the optimum, it would be bad if you're above the optimum. So necessarily, this is, you know, involving epistasis. When I say it's impossible for stabilizing selection to act independently of more than a few traits, that's simply because if every trait that we look at has a strength of selection of this value, which is often assumed in theoretical literature, then you're losing a few percent, you know, 2.5% of fitness per trait. So you just can't have that many traits, because if all those traits were varying around the optimum, and if there was quite a strong drop-off of fitness around the optimum, then you just be too unfit, you'd be dead. So that's a completely straightforward argument. I think it's actually the strongest argument here. Just to very briefly summarize some other observations, we know that, you know, selection experiments are often quite replicable. Sorry, yeah. Sorry? Okay, so you have to distinguish epistasis for trait and epistasis for fitness. I'll throughout be assuming that traits are pretty much additive, and that's simply because if we measure variance components for many traits, it is largely additive. And also, just for tractability, I mean, one can, of course, model all kinds of things, but we don't even have a good measure for the non-additive contributions to traits. You know, there are models, but they're not much empirical basis. And of course, even if you have an additive trait, fitness variation is, of course, epistatic, because it's the quadratic function of the trait, okay? And I'm not sure whether you'd count that as general epistasis. The epistasis is induced simply by the relation between fitness and trait, which is nonlinear, that's all. Okay, so replicability, if you do a selection experiment and replicate it and do a reasonably large selection experiment, you're often quite struck by how parallel the responses are. This is starting from a population which has variation, has standing variation. So that implies that there are a lot of genes involved, right? It's not going to depend on quirks of which genes coming. It's also not going to depend on mutation providing major genetic contributions. So replicability is quite striking, gives you some hope for the theory, actually. Also, some experiments, quite a few experiments, have selected on a trait and then relaxed selection, removed the selection. And very often, at least if you use a fairly large population, very often there's no return back. And so that suggests that there isn't, that you're not pulling up deleterious mutations which affect the trait in a positive direction, but which, when you relax selection on the trait, will pull you back. There are counterexamples. There are selection experiments which have picked up recessive lethals. And obviously, you can't fix those. You remove the selection, you go back down again. But again, typically, and I think somewhat surprisingly, you select on a trait, you shift the mean, you relax selection, and it kind of stays there. Right? VM, we've heard a lot about. The mutational variance increases by 10 to minus 3 or more times the environmental variance per generation. We have some idea now of the total mutation rate. We know very precisely the total mutation rate per genome, estimating the fraction of mutations that affect function, that are selected against primarily. That's harder, but you can look at the fraction of genome which is constrained, non-synonymous sites, for example, regulatory sites that are constrained, that evolve more slowly than the neutral rate. And that gives you figures approaching 1 or 2 for humans, for mammals. And I think Mike, in his talk, gave a slightly lower figure than I'm giving, but it's up of order 1, let's say, for human functional mutation rate. So quite a high total rate of mutation. But of course, we know that the mutation rate per classical genetic locus is small, 10 to the minus 5-ish, that order, depending on the size of the locus. So we know about mutation rates, more or less. Distribution of fitness effects. There's been a lot of work on the site frequency spectrum. Again, we've heard a little bit about this. If you look at the distribution of frequencies of alleles, which are present in 1, 2, 3, up to n copies in a sample, there's a prediction from the neutral theory, which is basically 1 over number of copies for the unfolded spectrum. And you can compare that with what you observe. And if there's an excess of rare alleles, that suggests alleles that are kept rare by selection. And you can get some kind of estimate, particularly from Drosophila. There's a nice review by Brian Charlesworth on this. You get some estimate of NES, assuming a sort of mutation selection balance. And these suggest that NES is quite large for a lot of mutation, a lot of deleterious mutation. That's also coming from classical experiments by people like Mukai, and I think we heard about the Bateman-Mukai type estimates before. Recently, there have been quite ambitious studies trying to jointly estimate through effects of linked selection, both positive selective sweeps, which produce sort of localized regions of reduced diversity, versus background selection, which reduces diversity rather evenly in regions with a high gene density and a low recombination rate. And one can try and fit models that estimate the relative influence of background selection against deleterious mutations versus selective sweeps in shaping the neutral spectrum. And these give estimates, but they seem to be a bit all over the place. So I haven't attempted to summarize that. Maybe they'll converge at some point. And then we also heard about the Buffalo and Coop type method, going back to an idea from Robertson, which tries to detect overall heritable variance in fitness without attributing it to a particular cause. So we have various sort of rather indirect ways of getting at the selection on molecular variation. Another thing which I think has been briefly mentioned, I think I mentioned it at some point, is that you can do something very simple, which is to turn off selection. And this was done by Alex Kondrashov in a paper with Shabalina et al. In what he called the middle class neighborhood experiment. You take pairs of flies, choosing them at random. You take 2 offspring from those, a male and a female, take 2 offspring from those entirely at random, so there's no selection. right? In principle, you've removed selection. And this is, of course, what's done in various different ways for mutation accumulation lines. What you find is that fitness measures such as viability and fecundity decline over time, and in this experiment by 1 or 2%. So that gives you a measure of the total rate of mutation times the average selective effect of each deleterious mutation, suggesting that deleterious mutations have, on average, and of course there's an enormous spectrum, a very wide range, but they have, of order 1% or a bit less sort of effect. And then finally, what you'd like to know is the number of traits that define an organism. But I put question mark there, and this is something we'll come back to because I don't even know really how to start thinking about that. Yes. What is our dimension? So those are the sort of observations, which a lot of them kind of intuitive, and we don't really know the precise values of these things, but we know the ballpark values. Yep. Yes, you can just go up here. Well, not necessarily low. I think all these numbers are surprisingly high. So total rate of deleterious mutation per diploid genome is just counting the numbers of new mutations that appear in an offspring when you've sequenced the parents. So you just take trios as the most basic thing, sequence the mother and the father and the offspring, and count the new mutations. And you get a number like that. Yeah. Sorry. Yeah. I should cross that out. That's a typo. Yeah. That's a confusion. Sorry. That was a mutation. Exactly. Yeah. Sorry. Yeah. That makes sense. So the fraction that affects fitness is lower. Yeah. You're right. Quite right. It didn't make sense as written. Yeah. Okay. So what I'll do is first of all just go through fairly quickly the effect of stabilizing selection on genetic variability. And I should say that throughout everything that follows, I'm going to ignore the non-genetic variants because it's kind of just annoying. And actually what we can do is think of fitness as a function of breeding value averaged over the random non-genetic variants. So we say an individual with a certain genetic component and breeding value has this fitness on average. Basically, of course, if you really want to practically apply any formulae, you want to include VE, but it really doesn't change anything qualitatively, so I'm just going to leave it out for simplicity. So what I'll do is look at the effect of stabilizing selection alone under various assumptions, then look at the equilibrium between stabilizing selection on a single trait and mutation, and then look at what happens with multiple traits, okay? And then we'll have a photo. So first of all, sticking with the infinitesimal model, it's really I find quite remarkable that actually Galton, in his 1877 paper, in which he remarked that the variability of the offspring was independent of the value of the parents, he actually calculated the equilibrium then between stabilizing selection. He had this model of Gaussian stabilizing selection versus the regeneration of variants by segregation of alleles amongst the offspring. So he'd observed that if you cross two individuals, you get variants among the offspring. He didn't understand VE, he didn't understand variance components, but he sort of had the idea that you released, or genetics, yeah, or anything. But but it's surprising he still got this calculation right. And then he assumed that it was good to be somewhere in the middle and bad to be extreme. So he had this sort of Gaussian model. And if you put this together, it's actually a very simple kind of calculation that at the phenotypic level, the distribution, call it psi of Z star, the distribution after selection is just equal to the distribution before selection times the fitness, the relative contribution of an individual, divided by the mean to normalize it, right? So that's, a very simple kind of model, and this W of Z might be the survival or the fecundity or something like this, right? So that's our basic model of selection. And if you apply that here, and if you say that the distribution is initially Gaussian and the fitness function is Gaussian, it's a product of two Gaussians, and you just do simple algebra, and you find that the variance after this stabilizing selection is lower, it's decreased from VG to something which is a factor of VS over VS plus VG smaller, so it's a smaller value. And so then you can use this infinitesimal model which says that if you take this population after selection, and if then you randomly mate individuals, the variance amongst families, that is the variance of the average of two randomly chosen parents, is going to be half of that, half of this variance after selection, plus then the offspring have this extra variance V0, which is generated by recombination. And so you put all that together, and then you say, well, let's suppose selection is quite weak, in other words, VS is quite large, and you find that the genetic variance, if there were no selection, would converge quickly to twice V0. That's what we heard about 2 lectures ago. But it's reduced by some factor by stabilizing selection. Stabilizing selection shrinks the variance. And this difference, this shrinkage, is just due to LD, as I explained in the last lecture. And these LD are transient, they'll dissipate. So if you remove selection very quickly, variance goes back to where it was before. Okay? So none of this helps us explain the maintenance of variation. Of course, we can just say V0 was there at the dawn of time and it stayed the same. And there truly are an infinite number of loci, but we don't really believe that. You know, we only have this infinitesimal model really validated over 100 generation timescales. So what determines this V0? That's the crucial question. And then we have to get into genetics. V0, so you have two parents, you cross them, and the offspring are not genetically identical. They're genetically different. And the reason they're genetically different from each other is that in meiosis in the two parents, segregation of unlinked loci on different chromosomes or recombination within chromosomes, that generates new genotypes. So when I say recombination, I'm including segregation of different chromosomes. And that's V0 is generated not by mutation, but by recombination. Okay, so let's look now at a model which is perhaps not that familiar, but is actually a rather general model, introduced, I think, by Kimura originally. So what we'll do now is focus on, not on the phenotype or on the breeding value, but on the contribution of a particular gene to a trait. So we imagine we take all the genes in a population, let's say 2 N genes in a diploid population, and we ask what's the contribution of each of them to a trait. And of course, we usually think of, you know, P and Q, 2 allele models, that's just for simplicity. But in reality, there may be a whole continuous range of alleles. And in fact, if you look at the actual sequence variability at any locus, there will be an enormous number of different haplotypes. Okay? So if those haplotypes affect the contribution to the trait, they may well do, then it may be more realistic to think in terms of a distribution of effects, right? And so what Kimura proposed was that at each locus, there's this distribution, and it has a variance little v. And the total genetic variance, assuming an additive model, is just twice 2 for diploids, plus the sum over loci of the variance contributed by each locus. Okay? So it's really straightforward. And the question is then, how does the stabilizing selection shrink that variance? And the insight to have is that if we just think of the effect of selection and we think of the mean being at the optimum, then we can just focus on the effect of one gene. Right? And the variance amongst the surviving genes will be shrunk by the factor that we just worked out. Actually, it's the same calculation we just had at the phenotypic level. And what we can do, just to keep it general, I'll actually start not by assuming a Gaussian. I won't make any assumption. I'll assume there's just some distribution. And we'll say that to a good approximation, if selection isn't too strong, the fitness is reduced by this 1 minus z squared over 2vs. So we have an actual fitness function e to the minus z opt squared over 2vs. And we assume z opt is equal to z bar. And we assume that z squared is much less than vs. And so then approximately, we can take the fitness as 1 minus z squared. Well, I take the optimum at 0, may as well. So we just have this reduction as you move away from 0, taking 0 as the optimal value. So that in there is our fitness. And if we want the variance after selection, we multiply the fitness by z squared, the deviation squared, by the distribution, and sort of integrate that. And we end up, you see here, the first term is going to be the average of z squared, which by definition is the variance. And the next term is going to be the average of z to the fourth, which I'll call the fourth moment, m4. So this is a fourth moment, average of z to the 4, over 2vs. And we've got this normalizing thing here, okay? And if we put this in here, we know that the mean has to be, this normalization, the mean fitness has to be 1 minus v over 2vs. And so what we actually have is 1 over w bar. That's our fitness times z squared times psi. And then we said that's 1 over w bar v minus m4 over 2vs. And then we say that w bar is roughly 1 minus v over 2vs. And then we can say 1 over 1 minus x is roughly 1 plus x, and we simplify it, and we end up with v minus so, you know, don't need to remember all the details, but the point is that without making any assumption about the distribution, we find that the variance has been shrunk by an amount that depends on this fourth moment. And that's an important thing, because we'll find that you get different conclusions about maintenance of variation depending on the shape of the distribution. And what I'll be doing now is going through contrasting a Gaussian model in which this fourth moment is just equal to 3 times v squared. That's just a fact about the Gaussian. Versus other models where there's a very fat tail, where actually what's happening is that we have a whole lot of rare alleles of large effect. And then this fourth moment is much bigger, and the reduction in variance, the effect of stabilizing selection, is much greater because you're eliminating these large effect alleles, large effect relative to the variance of this locus, not necessarily large in absolute terms. And so we get a different answer. So the point is that we can do the calculation using this machinery for the particular assumption that we have a Gaussian, and that's what Kimura assumed. But as we'll see later, that's actually biologically implausible. Okay? So really, I'm going through this contrast between sort of Gaussian models and rare alleles type models. So anyway, if we assume a Gaussian now, and then sum over all the loci, we find that summing this, sum of all the little contributions over all the loci times 2 for diploids gives us Vg by definition, and the sum of this thing, write it out, where are we? Yeah, so yes, so we've got a reduction, assuming the fourth moment is 3 times the seconds over v squared over vs. Okay. Yes, so that's this. And then we don't actually know what these v's are. They might vary from locus to locus. But we can do a little trick, which is to say, well, if the v's were the same across all the loci, then we have the sum of v squared would be n times the typical v squared. And we could divide by the typical v times n, etc. We can define an effective number of loci here. You can see that if all the v's were the same, then this would be nv squared, and this would be n squared v squared, 1 over n. If the v's vary, then this effective number is lower than the actual total number of alleles. But you can sort of use this trick to define this, which says that the genetic variance is reduced by an amount Vg squared divided by 2Vs divided by the number of loci, the effective number of loci. And then the crucial thing we'll come back to is that this is assuming a Gaussian distribution. If we have a fat tail distribution, the reduction is greater than this. Okay? So we'll come back to this. This is the continuum of alleles argument. It's a very general argument. But, sorry, yep. Yes. Lurch's zeta meets the abdominal bristle. That was the paper. Yeah, no, I'll come to that. I'll come to that. So the point, yes, no, I'm getting to that. So the reason I'm going through this continuum of alleles model, this is general. I'm then assuming a Gaussian, and then we get something, and we'll come to a prediction with the Gaussian, which all looks good. That was what Lande produced. Then contrast it with what comes out of sort of 2 allele models, or out of models which assume a much more fat tail distribution. And these ones, which Torelli argued for, are actually, I think, consistent with the data. So I'll come back to why we believe we can distinguish between these. The point is that different assumptions about the nature of this underlying distribution, which of course are very, very hard to observe, but different assumptions give you radically different predictions about maintenance of variation. So this is the continuum of allele way to look at it. I'll now do something which maybe I should have started with, which is the perhaps more familiar 2 alleles version, and we've actually seen these equations before. But suppose we, just take it simple, we have 2 alleles frequencies Q and P.
Can I ask a question here?
Yep.
Yeah. Just if you can just slow down a little bit.
Oh.
I was a little bit lost above.
Yeah.
I suppose I don't know if I'm the only one.
Which one?
But I was lost in this line 11.
Ah, okay, yes. Yeah, I'll go through that a bit more slowly. So, okay, so what we've got here is an argument about the little v's, which are the variances at a single locus. So you have some variability of effect of a single locus on the trait, and it's shrunk by a certain amount by this factor. And we then assume something about the 4th moment, and we say that it's shrunk by an amount here, right? So then we want to add that up and say what it is it overall? Well, we have Vg is equal to twice the sum of Vi after selection, which is equal to twice the sum of Vi minus, is it the factors of 2, over Vs, yeah. Okay. And then we say, well, this sum is just Vg. So then we've got to so let's say that, let's keep it simple and say little v is equal to Vg over 2n. Let's say all the v's are the same. That's probably the simplest thing. And then we're going to have this as equal to Vg minus twice. Now we've got n terms that are all the same. And each of the terms is Vg over 2n squared. And we're going to divide by Vs. And so then this is equal to Vg minus Vg squared. Now we've got n, we've got 1 over n squared there. So we've got n on the bottom, got Vs on the bottom, and we've got 2 on the bottom. So hopefully that's the same answer. So if the contributions were the same across all the loci, we'd have basically this, with this the actual number of loci. And all I'm doing here is saying, well, even if the v's vary, we can define an effective number of loci, which is a bit smaller, and plug that into there. So this will then be true. So this n is really the effective number counting those loci that actually contribute variance. You might have a whole lot of loci where v is very, very small, so we can ignore them. Okay, is that? Yep. Okay, so now we go to the 2 allele thing, which may or may not be simpler, matter of taste, I suppose. I just want to mention Wright's selection gradient formula, which is a really neat formula, which says that we can work out, you've seen before that we can work out the change in mean of a trait as equal to the additive genetic variance times the gradient of mean fitness with respect to the trait. So this is actually a phenotypic version of Wright's formula that I think was first introduced by Landy, and that was delta z bar is equal to this, VA times d log, well, okay, I'll do dw bar by d trait mean times 1 over w bar. That's one way to write it. And this whole thing is called the selection gradient beta. So that's a quantitative genetic way of looking at it. And Wright actually introduced this formula first for allele frequencies and showed that the change of allele frequency is equal to PQ over 2 times the gradient of log mean fitness with respect to allele frequency. So this just says if you increase the allele frequency by a bit, then that is what drives the change of allele frequency due to selection. It's the change in fitness due to an increase in allele frequency is a measure of the selection coefficient divided by 2 for diploids. It's just a diploid thing. Because mean fitness actually counts the effects of 2 genes in the diploid. So this is actually useful because if you want to find how selection acts on a particular allele that underlies a trait, and if we know that the mean fitness in this case only depends on the mean of the trait and the genetic variance, so approximately for this Gaussian fitness function that I keep rubbing out, the mean fitness is reduced by deviation from the optimum squared over 2vs and by Vg over 2vs. These are the two, if you like, loads, two reductions in mean fitness due to deviation from the optimum and variance around the optimum. Okay? So then we say that the selection coefficient on allele depends on two components. One is the gradient of log mean fitness with respect to the mean times the gradient of the mean with respect to the allele frequency, plus the gradient of log mean fitness with respect to genetic variance times the gradient of genetic variance with respect to dp. And this is just calculus. I think it's the chain rule, probably, something like that. Is it? No, maybe it's the, who knows what the rule's called? I've forgotten. Who remembers their school days? In calculus, when you have, okay, is it the chain rule? It's something like that, d by dx of some function of y of x. Yeah, chain rule, okay. Good. So, you know, we apply this basic calculus thing. And so this term here is the gradient of log mean fitness with respect to the mean. If you just do the calculus, you find the derivative of this square, blah, blah, it gives that. And then this alpha, that alpha over 2 is the gradient of the mean with respect to allele frequency, because we have that z bar is equal to twice the sum of alpha I pi. So the change in mean with respect to a change of allele frequency is just twice the effect. And we also have that Vg is equal to twice the sum of alpha squared pi qi. And the derivative of p times 1 minus p times q, is equal to 1 minus 2p, which is q minus p, and so on. So basically, these two terms correspond to the selection coefficient on the allele due to directional selection, which will only be there if there's a deviation of the mean from the optimum, plus this term, which is always there, which is due to selection against genetic variance. And we actually saw this formula before in a slightly different notation, and here I'm just identifying these two terms as basically directional selection, stabilizing selection. And actually, if you remember basic popgen, this term looks like selection against heterozygotes. So in a sense, as long as the mean's at the optimum, the selection acting on an allele will be entirely through the fact that the allele is increasing variance, and so an increase in heterozygosity will reduce fitness by alpha squared over 2vs, and so effectively you have a selection against heterozygotes. Okay. Bruce is warming up.
That's a really good point worth making, because it's kind of counterintuitive, but basically it's selected under dominance. So if you're sitting right at the mean, then if you fix one or the other, it's bad. If you're away from the mean, you can move towards it. So people often think if you have stabilizing selection, well, that's got to generate overdominance. That is, the heterozygote is advantaged on the underlying loci, when in fact, if you're sitting at the mean, it's actually underdominance, which is kind of counterintuitive.
Yeah. It's counterintuitive because if you think of a single locus in a diploid, you've got homozygote, heterozygote, homozygote, and the heterozygote will be fitter under stabilizing selection. But it turns out that if you've got a lot of loci and an additive trait, at most one locus behaves as if it's overdominant, almost all the other ones behave as underdominant. And Saul Wright actually showed you can maintain one polymorphism at one locus out of all the loci by a kind of overdominance, but essentially all the others typically are behaving as if they're underdominant because selection is acting to reduce variance. Okay, so we'll come back to this. So now we've worked out under various different models how stabilizing selection alters the allele frequencies or alters the variance contributed by locus, and we can just put this stuff together to find the equilibrium between mutation and selection. So Kimura actually, first of all, considered this equilibrium solution, and from before we found that at a single locus, the contribution little v to the genetic variance, little vg, is going to increase by an amount little vm, which is the mutational variance at that one locus, which is just mu times alpha squared if the allelic effect is alpha. And then it's reduced by this amount we had v squared over vs, right? And you can just solve for the little v. Say at equilibrium, we find that the variance contributed by this single locus is just root vm vs, right? So it's a really simple formula. And we also know that the total mutational variance that we can actually observe is for diploids 2n times vm, where n is the number of loci. And then if we stick this together, we multiply this by, let's go through the thing. So we just again sum over all the 2n genes. Little n is the number of genes contributing. So we've got Vg is equal to twice sum of I of square root Vm times Vs. Is that right? Yeah. But Vm is equal to the total Vm divided by 2n. And then if we assume everything's the same, then this sum is just times n. So this is equal to the square root of 2n Vm Vs. So now we've got a formula which is based on things we observe at the level of the whole trait. And we've got, I've sort of simplified things by assuming everything's sort of equal across loci, but it's not too much of a problem. So we've got, a simple formula that depends on the number of loci contributing, the mutational variance, which we know, and Vs, which we kind of think we might know. And if we plug in the traditional choices for these, we find that we don't need that many loci. You need 25 loci, 2 times 25 times 1 times 20 VE gives us Vg equals VE, right? So this was an argument made by Lande in a 76 paper, which is very influential, in which he said, actually, given what we know, then we can maintain observable high heritabilities, 50% heritability, Vg equals VE, with 25 loci, so that seems fine. And in fact, Lande developed this. He developed it to multiple loci to include linkage disequilibrium, and he really pushed the maths because if you assume a Gaussian, you can do anything, you can solve anything, kind of. And he applied it to study all sorts of problems like sexual selection and clines and speciation and pleiotropy and so on. So it was very, very innovative and very ingenious. However, it depends on the assumption that this distribution of allelic effects is Gaussian. Any questions? Yes. Do you want to go back? Yeah.
Can you explain again what Kimura contributed? How he got to this square root? I'm sorry.
Yes. No. So what Kimura did was actually more elaborate than I've described. He actually used a diffusion approximation, which is what he was sort of famous for developing. And he used it to suppose, basically had a model where you have a distribution of allelic effects you have stabilizing selection acting on that, and just thinking about one locus here. And you have mutation, which he actually put in, and the crucial assumption was that it was kind of diffusion, so mutations were a very small effect. And so what mutations were doing was just adding a little bit to the variance each time, so the variance would be increased in a kind of diffusion. He wrote down the diffusion equation and showed that if the mutations coming in were of small effect, basically small relative to the standard deviation of this distribution, then you would get a Gaussian. And so it was kind of a beautiful thing. It would be beautiful if it were true. Look, we don't actually have measurements in any of this. I mean, having said that, I mean, I think it might be a plausible model for things with high mutation rate like microsatellites, where you can imagine, and we know about the mutational process, you have a number of copies, you have 40 copies, let's say, of a repeat, and mutation slippage are ±2 or something like that. So that's a measurable mutation model, which is rather similar to what Kimura assumed. So Kimura basically contributed, the idea of the Gaussian model, the idea of looking for mutation versus stabilizing selection, and the demonstration that under certain assumptions you will get a Gaussian. That's Kimura's.
And what you're saying, this is for a single gene?
This is for a single gene, and then you just add up over all the genes.
And so you get a distribution of effects, which means that you have a large number of alleles?
Yes, that's crucial to the continuum of alleles. You have an infinite number. So this is all deterministic. There's no drift in any of this. So you assume an infinite number of copies of the gene in the population, and they might all have a different effect. You know, it could be true. If you look at a microsatellite, that's, you know, not far off. You've got a whole range of effects, which is kind of Gaussian. So you have to have many alleles, yes. I mean, Monty Slatkin had a neat paper showing that 5 alleles was plenty. You know, he did 2 alleles, 3 alleles, 5 alleles, and, you know, you pretty quickly get to a Gaussian type solution, but you need more than 2, yeah. Okay. So, you know, Kimura and Lande between them produced this Gaussian model and made a prediction and said, you know, actually we can put in plausible values and we can explain heritability. If you do the 2 allele version, and this was solved by Latter, I think, first, and then developed by quite a few other people after that, you have this formula for selection, and you just add in some mutation, and this is symmetric mutation where, you know, the Q alleles mutate at rate mu into P, and the P alleles would mutate into rate mu into P, and so, sorry, into Q. So you've got, you know, standard symmetric mutation rate mu in either direction. Doesn't really matter about the symmetry assumption. And then actually you say, well, set that to 0 and solve it. And it's kind of complicated actually, but you can wave away the complications by saying, let's suppose the mean's at the optimum, which usually it will be, so that's not too bad. So if the mean's at the optimum, you cross that out. And now you can see you've got 2 terms, and you will get a solution if P equals Q. There'll always be a solution at the symmetric state. It's a symmetrical model. So P equals Q, that will give you 0. And that's fine, because basically you've got an effective underdominance, and you'll sit at this unstable equilibrium, not changing if P equals Q, and mutation won't change if it's symmetric. So you'll have a solution at P equals 1/2. But it turns out this is only stable if mutation is stronger than selection in this sense, if the allelic effect is small relative to mutation rate times Vs. So if mutations have an effect larger than this threshold, then you can cancel out the P minus Q term, and you end up with alpha squared over 2Vs as PQ equals mu. I suppose I can write that. So you end up with mu equals alpha squared over 2Vs PQ. And this is basically the standard mutation selection balance from Haldane. You've got a mutation rate mu, and you've got an opposing selection alpha squared over 2Vs, and those will balance each other when PQ is equal to mu, 2Vs mu over alpha squared, okay? So that's just mutation selection balance, where the selection coefficient is this selection pushing the heterozygotes down because they're inflating the variance, and that's bad. And so the VG is just equal to a sum There's about to be a miracle, because this alpha squared cancels with this alpha squared, and we end up with 2bs u over, well, the alpha squared's cancelled, and this whole thing is equal to 2bs u, where u is equal to 2 times the sum of mu I. So we can actually have mutation rates varying, allelic effects varying across loci. And what happens is, as in the classical mutation load argument, alleles with a large effect, alpha large, they'll be rarer because the selection against them is stronger. And so Vg is a sum over alpha squared times pq, but pq has an equilibrium which is 1 over alpha squared. The alpha squared's cancelled. So regardless of the allelic effect, as long as it's big enough, you'll have this result which is independent of allelic effects and which depends only on the total mutation rate. This second term is just a bit trivial. It's the sum over all the diddly ones which have a small effect below the threshold, but that's going to be small. And actually, I'll sort of ignore this for the moment and just say that this U star is really the total mutation rate to loci that produce allelic effects big enough to be above this threshold. And this is something tangible, which we can't quite measure, but we have an idea of what the order of magnitude might be. And one can understand it actually in terms of this mutation load argument, where, as Haldane showed, if you have mutation selection balance, what? Question, sorry, yes. One can have enormous fun, if you like, doing this sort of thing, looking at the stability. Because this formula, if you solve the whole thing, I did this in 1986, and it was fun doing it. It's kind of, nerdy, hobby thing. So this is actually a set of coupled equations, because the z-bar, if you take into account the deviation from the optimum, this z-bar is a sum of alpha I, pi, right? So you've got these coupled via the z-bar, and then you can do the stability matrix, and you work out the eigenvalues and all this. And what you find is that basically there are a large number of stable equilibria which have roughly, alleles, the ideal state, if the optimum is in the middle, is for half the loci to be fixed for plus allele and half for minus allele. But actually you can have, 40% of them for plus and 60% of them for minus, and it'll still be stable. There's a narrow window in which you get stability, and it's all a bit complicated, but in the end what it boils down to is that all of the stable equilibria have the mean very close to the optimum. Selection is really effective at keeping the mean at the optimum. So all I'm doing is saying, let's say the mean's at the optimum, and in that case, these results are looking at the stability of the single locus result, which we can do. It's basically underdominance plus mutation. And if mutation's dominating, you're in the middle, and if selection's dominating, you're at the edges. Yeah. And so this PQ here, that actually masks the fact there are two alternatives. P could be small or Q could be small. So you've got a sort of system where either you're held in the middle by mutation, or you're at the edges with one or other allele rare, and that's the regime that's interesting, actually. Yeah. So one way to think about it is in terms of mutation load, because under classic mutation selection balance, regardless of the selective effect, as long as it's stronger than mutation. So, question? Okay. Now I'm on the lookout for any signs of questions. Okay. So with classic mutation selection balance, the loss of fitness due to mutation at a single locus in a diploid is just 2 mu. Okay. As long as there's some degree of selection on the heterozygote. And the loss of fitness we know in total is just Vg over 2Vs. That's our genetic load due to variation around the optimum. So it sort of has to be true that Vg over 2Vs, just from the mutation load argument, is equal to the total diploid mutation rate. Okay? So actually we could have got to all this just from a mutation load argument. So it's kind of neat. So we've got two all completely different kind of modeling frameworks. I mean, they're consistent with each other. They're making different assumptions about allelic effects. So just to try and give you a handle, and there was an endless well, not endless, but it seemed to go on forever. There were lots of back and forth papers about which regime was more plausible, and it's summarized in a masterful chapter in Walsh and Lynch, I think. Yeah. Anyway, yeah. So the Gaussian continuum of alleles model, the Kimura-Lande model, requires that the effective mutations be small relative to the standing variation at that locus. And that requires that alpha squared is less than, little v, which is less than root Vn Vs. But we know that Vm, the mutational variance per individual locus, is mu alpha squared. So we want to have, for this to hold, alpha squared less than so that's the mutational variance coming in, mutation rate times square of the effect times Vs. And so if we square both sides, we have to have alpha to the fourth less than mu alpha squared Vs, or alpha squared less than mu Vs. Okay, so we've now got a constraint on the allelic effect, and then if we plug in our favorite values for Vs and mu, per locus we're thinking mutation rate 10 to the minus 5 or something like that. Then we have to have alpha, 0.014, pretty small. In other words, it's got to be 1% of the environmental non-genetic variance to be consistent with the Kimura-Lande regime. But then we say, well, you know that Vm is just 2n times mu alpha squared. We know what that is. So now we've got a constraint. We have to have at least 3,600 loci, right, in order for the effect of each locus to be small enough to fit the assumption. But we've already calculated somewhere up above that to explain heritability of around a half with this model, we need 25 loci. So there's a contradiction in there, right? So the point is that if we assume a really small mutation rate per locus, and we assume the effects of the alleles are so small that we're fitting this Kimura-Lande continuum of alleles thing, then in order to get Vm as big as we observe, we've got to have an enormous number of loci. But then we'll have an enormous genetic variance. So the whole thing doesn't make sense. And there's different ways of playing all these number games. But in the end, the Gaussian regime seems feasible only if the loci involved have a very high mutation rate. And in fact, the Torelli 84 paper, the wonderful Lurch's zeta meets the abdominal bristle, which was a triumph of at least the title anyway, but it was a theoretical paper plus a lot of analysis and review of the empirical data way back then, 40 years ago. Actually it wasn't obvious then, it probably isn't even obvious now, that all loci responsible do have a low mutation rate. You could imagine that if microsatellites and transposons and who knows what has an effect on traits, then maybe there are high mutation rate loci contributing. But it seems, and certainly from GWAS now, that there's a lot of loci, and they're each contributing a very small genetic variance, and that typically the alleles are rare. So we actually know directly now a lot about the relationship between effect size and allele frequencies, and it really seems to fit the rare allele version. It's not that you necessarily have to have two alleles, I've assumed that for simplicity, but you have to have a distribution of effects at a locus in which you typically have a common wild type, and then you have effects. Let's say this is one standard deviation, but what's driving that standard deviation is a whole lot of rare alleles which have really big effects of several standard deviations. These are standard deviations of the contribution of that locus. Okay, so that, yes. Sorry?
So I don't need to worry about continuum of these models.
Well, if you're especially fond of continuum mathematics rather than, and calculus and analysis, rather than clunky population genetics, I think you can still apply it. It is still relevant, for example, to asexuals, because in asexuals you have a single big genome with a very high total mutation rate and a continuum of effects. So actually, there is a whole body of literature now, it's kind of fitness wave type literature, looking at the evolution of the fitness distribution of an asexual population. And that is a continuum of alleles model. I think it's also maybe the case that the continuum of alleles model works or is reasonable for particular loci with high mutation rates. microsatellites and stuff. But in general, in the context of quantitative genetic variation due to many loci in a sexual outcrosser, yes, I go with Torelli and not with Lande. So, yeah. And I think that's probably the consensus in the field, but yeah. Laura. Microphone.
You have this result that the variance of small effect alleles is the same as the variance of large effect alleles.
Yes.
But in an infinite sites model, you don't have that result.
Oh.
And so I'm just wondering where this discrepancy comes from.
I think because this is deterministic. Whereas sites,
so when you take into account drift.
Yeah. When you take drift in, it's different. And then, of course, it's more complicated. You still do it. I mean, but you're looking at distributions of allele frequencies. Yes.
Yes, and then which we do expect drift to exist, and so you would when you do take into account drift, you actually get that the variance contributed from small effect alleles. I mean, it increases linearly with the effect size.
Okay.
For very small effects.
For very small effects, yeah. Bruce?
You can also think of the large and small effect variances being very similar as kind of Haldane's rule, where the selection coefficient cancels out, basically.
Yes, so it's coming from the deterministic mutation selection load argument, but that is a deterministic argument. And you put in drift and that breaks down. But I think that doesn't alter the qualitative conclusion that we're in the rare alleles regime and not in the continuum alleles regime. Doing drift with continuum alleles is quite a challenge. And so it's less, I mean, it's been worked out, but it's a more complicated thing. Okay, so I'll finish up by trying to introduce a bit of reality, not too much though, and that is to realize that organisms are more than a single trait. So all this has been single trait arguments, and it's sort of, you know, it seems reasonable that, you know, if we put in this, where was it, the rare allele prediction which we had, which was that we'll get rid of all this continuum allele stuff. We'll have VG is something like 2UVS in the deterministic theory. Well, what can we put in? If we put in the VS is 20VE and we want to maintain high heritability, then we need a total mutation rate to alleles affecting the trait of 0.025. We could imagine that. We know from GWAS that loads and loads of loci affect each trait, so fine. But the trouble is, what if we have more than one trait? And if we have separate sets of loci affecting each trait, and we have 100 different traits, which isn't that much, I don't think, then you've got a total big U has to be 2.5, and that's more than we could stand. So you can't have 100 separate sets of loci, each driving this mutation selection balance. But then say, well, okay, we have pleiotropy. So we have loads and loads of alleles. We have a total mutation rate of order 1 or 2. Let's say big U star is something quite big. And we say that all functional regions affect all traits. So fine. But what we'll see in a minute is that then the prediction is that the selection acting on each allele is the sum of its effects on all the different traits. So it's not just alpha squared. It's not just the selection coefficient is alpha squared over 2VS. It's the sum of all the alpha squareds. It's the sum of all the deviations from the optimum that it's causing for all the different traits. So if we have n traits, then the allele frequencies decrease by a factor of n, and we end up with a prediction VG for a particular trait of 2UVS over n. Another way to look at it is we have this load argument that the total load summed over all traits is 2U, and so the load attributable to one trait has to be of order 1 over n of that. So, you know, we're really stuck if we want to have more than, 100 or so traits under strong selection. VS is this. So now we're kind of in trouble. Now, there's a whole different modeling tradition, which in some ways is much more straightforward. And I will trace this back to Trudy Mackay in sort of 87, I think, who did a neat experiment. In those days, the old days, it was hard to, there was no CRISPR or anything, but she had the ingenious idea of throwing transposable elements into random locations in Drosophila and looking at the effect of a transposon insertion, random insertion on a trait. And as everyone knows, Drosophila only has one important trait, the bristle number. Actually, it has abdominal bristles and sternopleural bristles, right? So that's what people measure. So she looked at the effect on bristle number and on fitness, or rather viability, as a proxy for fitness. So you have a joint distribution then of a particular mutation of alpha and S, and she sort of measured this joint distribution. And you tended to find that transposons that had a large effect on the trait had a large effect on fitness. And then you could imagine sort of just some very simple modeling where actually the trait is kind of neither here nor there. It might be a little bit selected, but let's just ignore that. And let's say that, look, you've got alleles coming in, and they have an effect on fitness S, and they have an effect on the trait alpha, and what matters is this whole sum. Okay? And actually, if you look at that sum, it's just Vm over S, all right? That's not quite the case, because you could start worrying about the covariance between the mutation rate and the selection coefficient and the alpha, which is kind of what Trudy McKay was measuring. But naively, if we just said there's an effect on the trait and there's an effect on fitness, and they're sort of independent of each other, then you get this Vm over S, which I think we went through in the class last Friday, the exercises. And all that's saying is that, well, if you've got a certain standing genetic variance, you've got mutation coming in, increasing the variance by Vm per generation, you've got to have something eliminating that variance. That is, the underlying alleles have got to be eliminated at a rate S to match that. And so this is actually a very robust result, which doesn't depend on that many assumptions. It's really about the relative timescale of mutation coming in, increasing variance, selection, eliminating variance. In fact, if you have a distribution of alpha and S, which in principle is measurable, then it's the expectation of alpha squared over S that matters. Now, you can look at it another way. It's actually rather, and I'll sort of be working towards that. But suppose we say, well, the trait does have an effect. We care about the trait. It affects fitness. It's understabilizing selection, Vs. And so now we have basically a selection coefficient S due to this sort of extrinsic deleterious effect, which could be anything, plus alpha squared over 2Vs, which is the selection against increasing variance in the trait. So now we have this formula. And what you find then is that depending on whether this is larger, typically, then you get this Vm over S result. Or if this is smaller, then you get this 2UVS result. And the actual total variance maintained is actually less than either of those. So we've got some sort of handle on this. And regardless of that, the net selection, this plus this, must be of order 0.001 or 0.01, because that's the rate at which variance is being eliminated. So that's the case, a really pleiotropic model in which you're just saying, mutations come in and they have an effect on fitness, they have an effect on the trait, and the effect on fitness is what drives their frequency. But another way to look at it is to think that, well, actually we have multiple traits, that this S is actually due to effect on all these other traits. So now we have a subtly different model which says there's loads of traits, each under stabilizing selection. So we have a sort of multivariate quadratic thing. And sometimes people call this Fisher's geometric model, multivariate stabilizing selection. And we replace this S by basically the sum of the alpha squareds over 2Vs for all the other traits. And so we end up with this formula, which has a sort of intriguing consequence that the genetic variance for a particular trait doesn't necessarily bear a direct relationship to the selection on that trait. I'm assumed here that Vs is the same for all the traits, but you can do the same thing with Vs varying across traits. And if you say that we've got stabilizing selection on a certain trait that we can measure and observe, it may be that the genetic variance on that trait is due more to the selective effects of the underlying alleles on all the other traits. And in fact, Simons et al. Have made a model of stabilizing selection on very many traits using this sort of Fisher geometric model framework, using this sort of multivariate quadratic framework. And they find that in the limit of large n, under the assumption that mutations have a random direction of effect, they will have an effect on some set of traits which is random in direction. And then you find that there'll be some relation between the alpha 1 squareds and all the other sigma alpha squareds. Basically, a mutation with a large effect on traits in general, that is a large radius, will tend to have a larger effect on your focal trait. but it'll be very stochastic and random. So there's a somewhat loose relationship. But they can predict the joint distribution of allele frequencies and effects, and that's something testable, and they claim it fits. So one can make progress with this, but I just want to finish by saying that actually there's a really subtle difference between what I've called the pleiotropic models, which are in this sort of Trudeau-Mackay, framework, Bill Hill framework, where you say there's an effect on fitness, which is just straight deleterious, versus the multiple traits version, where you say, well, it's due to deviations to the optimum in a huge range of traits. And the difference is that if an allele is straight deleterious, then if you fix it, you're stuck. You've got a permanent load. You can't compensate it. Whereas under the multiple traits thing, if you fix an allele that takes you from the optimum, another allele can take you back towards the optimum. So there is a real difference in the long term for population survival about whether or not you allow compensatory mutations. So this framework, although it gives similar algebra, really, and similar predictions for maintenance of variation, it does have different consequences for the long-term survival of the population. Yeah. Well, okay, it's a matter of assumption. So here, let's say I had at the back of my mind that these were just straightforwardly deleterious mutations. There will be such mutations. If you remove hemoglobin, then that's bad, right? So the model here is that you have alleles coming in which, regardless of the genetic background, reduce fitness by S, and that's it. And there will be, if you put drift in, there's some chance they'll fix. And so under this pleiotropic model, you'll have a very slow decay of fitness. And of course, in reality, there is the possibility of compensation, but it actually isn't obvious to me that we can always be saved by compensatory mutations. I suppose the fact is we have been, but it's a slight worry. Okay, so I'll just finish with a summary. Yep.
Can I ask a question on the last equation?
Yep.
How does this still has the problem that you were mentioning when you have many traits, independent traits that
we haven't avoided that. We've still got that problem.
Okay.
Yep. So I'll come to this in the summary.
So the idea here is just that selection coefficient will depend on the effects on all the traits, basically.
Yes. So the selection coefficient, which determines the allele frequency of a particular locus, will depend on its effects on all the traits in the organism. Yeah. So anyway, summarizing, firstly, it's complicated, which I think everyone has gathered, good. So assuming a balance between mutation and stabilizing selection, even the simplest models depend on the details, depend basically on the shape of the distribution of effects. If you have plausible per locus mutation rates, the causal alleles are likely to be rare, and they'll have large effects relative to the standard deviation of that locus. And so we're in this sort of regime where we expect the standing variance to be dependent on the total mutation rate to alleles affecting the trait. And so then we have a problem, which is that we don't know, we don't understand really how large numbers of traits can be maintained like this. And I think really the resolution is that, well, maybe the resolution is that it's kind of stupid to assume VS equals 20 VE, assume strong stabilizing selection on everything. If we take an arbitrary trait, we do measure strong stabilizing selection because it's correlated with a small number of kind of leading principal components, which are important. And it has to be the case that very large numbers of traits are maintained near some kind of optimal value. We're complicated, things need to function from an engineering point of view. I think there have to be large numbers of traits under stabilizing selection. But that stabilizing selection has to be very weak on most traits. You can't assume strong stabilizing selection on an indefinitely large number of traits. But regardless of all those arguments, one thing I think we do know is that the typical selection acting to eliminate variance is of order 10 to the minus 3-ish. So that sort of gives a little bit of hope that it might be almost measurable, I don't know, in sufficiently heroic evolving resequencing experiments. So I'll stop there. Yeah, thank you. Does anyone have any other ideas for how to explain variation? I must say, all this is very narrow-minded. It's all very much focused onto mutation, selection, balance. There is other stuff going on. There's balancing selection, which could be contributing. There's fluctuating selection, spatial variation, who knows what. But
So I think a lot of the discussion is in the literature is motivated by ecologists that focus on a particular trait, wingspan or beak size, when in reality, selection is operating on some fuzzy, highly multidimensional phenotype, and you're probably not getting just selection on your trait, you're getting a projection from this highly dimensional surface onto your trait that then you score as having some effect. And so I think a part of it is this fact we typically think we meaning like field ecologists, think of a few traits as being important for fitness and focus on those, when in reality, selection is operating on some multidimensional structure, really have no idea what it is.
Which is very unhelpful if you're empirically trying to measure selection. Right? Yeah. Solve what? I mean, yes. Well, I mean, you can solve anything by simulation or numerically, but actually, I mean, the maths is quite simple. I mean, it's a quadratic surface, and so Simons et al. Get quite a long way in working out this joint distribution of whatever. Yeah, I think the bottleneck really isn't the mathematics or the numerical computation. It's actually trying to find things which give a robust prediction about observable quantities. Yeah. Yes, I mean, once you get a continuum of alleles with drift and different kinds of selection, then, you know, it can be a bit of a pain, but it's just physics, which is simple. So, I mean, one thing I want to mention is that, you know, when I said this sort of straightforward theory predicts that there shouldn't be much relation between the genetic variance for a trait and the strength of stabilizing selection on the trait. And there's not a lot of data on this, but I guess one would feel that there should be. And there is a literature which asks, is there evolution of modifiers which increase the canalization, reduce the variance, either environmental or genetic, of traits that run strong stabilizing selection? And so that is a whole extra body of theory involving epistasis and so on.
What the human people have done is you can use the joint effect frequency size for a number of traits and then estimate some general selection coefficient averaged over all of those. And when you do those over a large number of traits, the results are rather remarkably constant, which is kind of surprising.
It's kind of weird, isn't it? But maybe supports the idea of plyotropism.
Yeah, I mean, there's some exceptions like schizophrenia, but why would body mass index and some behavioral trait have a comparable? Yeah. And that suggests, again, you have this very high dimensional fitness structure that you're just seeing projections down onto the dimensions you're looking at.
But that maybe is just a modern version of the observation that VM is rather constant across traits and VG is constant across traits. I'm not constant, but the same ballpark. Yeah, sorry. Sorry, yes. Yeah.
Do you mind explaining a bit more about this third conclusion on why exactly can't we have, like, what is this large number of traits that wouldn't be able to be understabilized and such?
Well, this is actually a completely straightforward argument that if you imagine, you know, without going into any of the genetics of maintenance of allele frequency variation or heritable variation, if you just take the idea that there's VS acting on a large number of traits, for each trait you lose fitness VG over 2VS. And so if that VG over 2S was 1 over 40, which is the kind of assumption that was made in the early theory, and I would say that's moderately strong stabilizing selection, which, you know, is observed for typical traits. That's what, you know, it's not that it's completely baseless. But you can't have that strength of stabilizing selection acting on huge numbers of traits because you'll be losing fitness through your deviation from the optimum in all those traits, and it would add up. So the idea is that you have a projection that there is some principal component, or maybe, you know, 20, 30 principal components, which are metabolic rate or body size, which really, really matter. And then if you measure any morphological thing, it'll be correlated with that, and it'll pick up some of that strong selection. But the things orthogonal to that, which might be in the tens, hundreds of thousands, might be under very weak stabilizing selection. And then it's kind of fine. It'd be nice to have some way of measuring that. I don't know. People have tried to estimate strength of stabilizing selection on gene expression. which is a large number of traits. But Bruce, what do you make of that literature? Messy, okay. Yeah, that's the kind of thing one would like to be able to do. That's an interesting thing. I think I feel guilty at producing a lot of very misleading theory early on, assuming equal allelic effects where that's the case. Where if you have equal effects, you can't quite get to the optimum and then you have variance on. But actually, if you put in a distribution of effects with big effects and small effects, you can, it's like doing the integers, right? You add up to any arbitrary real number by having some big numbers and some small numbers. And you don't need much of a range before you get really spot on. You still have alternative equilibria, but they all look the same. So it's a good point, but I think you have to beware of simple-minded theory. Yeah. Laura?
When you say strong stabilizing selection, you mean big VS or small VS?
Small VS, yes.
So, but VS somehow isn't I mean, it's VG over VS we care about. Because if you change VS, then in an equilibrium model, VG just changes correspondingly. Right, because VG over VS goes like U over N.
I see what you mean.
And then if you try and think, like, how would you measure VG? It really just depends on the units you're using for your trait. So that...
Yeah, now you've confused me, yes.
I mean...
You're right, I think what matters is the total mutation rate in this way.
Yes, I don't think VS is a real parameter because your choice just determines...
It is a real parameter. If you're measuring a wingspan or something, you want to know what VS is.
But then VS is, then it's... Well, you either get VG over VS, right? Which depends on the, I mean, in an equilibrium model, I don't think it's a real parameter. Or you get VS in some units, whatever units you happen to have chosen. But that's not. So you're saying that VS over VE determines the strength of stabilizing selection? That's how you would measure it?
Yes, that's how it's usually represented.
Okay.
I know where you're going, Laura. You're in love with your scaling, which is which I can never get my head around. Anyway, good.